All right, guys, so welcome back. And so now we've aligned our sequences to the human reference in our case. And we've now moved from, let me grab my pointer. We've moved from our raw output, our FASTQ files that we know how to perform quality control on, determine, if they're telling us the right base pair at the right position. And now we're aligning to a genome. And this produces the SAM-BAM file format. And you can see all of the different file formats listed here at every step of our hypothetical pipeline that we're running through. And now we've aligned to our reference genome, and this produces the SAM file format, or the sequence alignment map format, and the BAM just being the compressed version of that. And so we compress it into BAM because these SAMs tend to be extremely large, especially considering, looking at the human genome and, the size of it. And so basically this looks sort of similar to our FASTQ files. Remember, the FASTQ files just mean a huge list of our identifier at the top, our gene sequence, and then, a bunch of information related to the quality that looks kind of like this very confusing information here, but this tells the computer, certain information about the quality of each base that was called. And so this SAM-BAM format kind of combines both the information from the FASTQ and information from the reference that we align to. And so you see here, we have our actual short sequence information, we have our quality information, but we also have, where this particular short sequence aligned to our reference genome, and we have that very, very long list again of all of that information. And so to perform this, we used a program called Bowtie 2. You can see there a image of the publication, and you can read about it more if you are so inclined. Of course, you can search on PubMed and go find this quite easily. So it's been well used in research and the medical kind of sphere, and we're going to be using it, or we did use it for our activity for aligning our whole exome sequencing reads to our reference. So we used Bowtie 2 to align sequencing reads and visualize the sequencing reads using a tool called IGV. Now in IGV, remember, we are going to see a number of what are called these SNPs, or single nucleotide polymorphisms. These are the most common type of genetic variation among people. Each SNP represents a difference in a single DNA nucleotide. For example, here we have 3 individuals. We have one individual with an A at that particular position in the genome, another individual with a G, and the third individual with a T. And so this is a SNP at that particular position with an AG and a T nucleotide. SNPs normally occur throughout a person's genome and DNA. They occur almost once in every 1,000 nucleotides on average, which means that an individual probably has 4 to 5 million of these, when you align your genomic sequence to the reference. And so remember that we were visualizing using the web-based version of the IGV tool, the integrative genome viewer. And if you didn't complete that exercise with SNPs 1, 2, and 3, go back to that individual lecture and go ahead and finish up those questions. And so remember, now we're performing, the alignment with bow tie 2, and so we see things like this when we actually align our short sequencing reads, which are indicated by these gray bars here. So each of these is one of those short sequencing reads that we got from the next generation sequencer. And if it's grayed out, it just means that it matches our reference sequence being up here. And so everything that's grayed out means that it matches the CAT, C, GG, CC that we see there. And anytime it actually calls the base, it means that it's different. And so when we get here, we see a G to T SNP, right, at that position, because at that position, our reference genome has a T allele at that particular position, and about half of our reads has a G. And so remember that we are diploid organisms or have 2 copies of each chromosome, and so it looks like in our case one chromosome has the G allele at that particular position and the other chromosome has the T, matching the reference. And so we are now taking our raw reads in the form of our FASTQ file. This particular pipeline wants you to use a particular sequence mapping tool called BWA, which is also a very well used option. Again, we used Bowtie 2 to produce our BAM files. We then need to take our BAM files and actually perform variant discovery or variant calling. And there are several programs to do this as well. And at the end of our variant calling, we get out what's called a VCF or variant call file. And so just to remind us of the variant call format structure and how we actually interpret that. So each line in our variant call file represents a single genetic variant that's different from our reference when we aligned those short reads. Right, we have chromosome and position, we have ID, so this is an optional identifier for a specific variant, and so you might see a lot of times what are termed RS numbers, so this is how NCBI actually curates all of the SNPs that it gets through every research-based project that's submitted to them. You have reference and alternate calls, so reference basically means this is the nucleotide that was found in the reference sequence, and the alternate is the nucleotide that was identified in the sample reads. You have quality scores that come next, so you can determine, if it's a call that's based in high quality. Any filters that you imposed on the specific variant caller. And then this info column becomes actually very important when we're doing annotation that we did right before break. And this is when we perform various, site level annotations. And we'll explain a lot more of this in the coming slides, but the info column is sort of a catch-all for any type of annotation that we might perform on that particular variant. So how it impacts a particular gene or exon, or if it's found in any other databases, what the proportion of that specific variant is in the general population. So we can add all of those things to the info column and while still keeping the structure of our VCF file. The format column, which is over to the right side here of our VCF. So you'll see a string of numbers here that's separated by a bunch of colons. And this gives us information on the specific call at that particular position, as far as quality of assigned genotype, the filtered allele depth, which we'll talk about a little later, and the genotype at this particular site. So where 0, 0, remember, means that it matches the reference in both alleles. 0, 1 is heterozygous, and so this 0, 1 here means that they had one copy of the T allele and one copy of the G allele. And so it would look very similar to us visualizing a SNP in IGV. So half of those reads roughly would have a G allele and the other half would match the reference and have a T allele. In the case of homozygous reference, they would have two copies of the reference allele. So whatever that happened to be. And then 1, 1 is homozygous for the alternate allele. And so this individual has two copies of the G allele at that particular position in the genome, whereas our reference sequence has two copies of an A allele. So just basically speaking, how does a variant caller work? And it's actually pretty, intuitive when you think about it. It's just doing it on a very, very large scale, right? So it's taking those short reads, it's aligning those short reads to our reference. So the short reads, because we have millions to billions of them, will have kind of a number of reads that match up to any particular portion of our genome. And so in this case, we have 5 particular reads, and this type of thing where we have a number of reads stacking up to our reference is known as sequencing depth. And so if I ever ask the question, what's the sequencing depth at, the G allele, the capital G allele here, you would say 5x, right? So there's 5 reads that are aligning to that particular spot in the genome. And so basically, what it does is similar to what we did, manually by looking at these SNPs in, a visualization software like IGV. So we evaluated and saw, okay, read 1, read 2, read 4, and read 5 all have a G allele at that position. Read 3 has an A allele. And so we then, so if we were manually looking at it, we would start to ask ourselves the questions, okay, this variant can be one of 4 things. It can be a true variant that was called that's actually biologically real. It can be an experimental artifact. It can be a base calling error, or it can be, an error in analysis or misalignment, something with the software that we used. And if we're not actually looking at it, manually, right? This is decisions that the variant caller makes on our behalf based on the inputs that we put into it. And so remember, this is what it looked like when we were going through IGV. So, it's very similar in that, it gives a confidence score based on that variant call that it made. And it allows us to evaluate, how sure it was that that is, an artifact, for example, or a real variation. It just does this on a very, very large scale using the power of the computer. And so remember our activity, we used a variant caller called Freebase to output our VCF files. And remember, as with every piece of software that we use, there are a number out there that that all perform very well. They, some tend to do better on other types of variation than others, and we need to know kind of the ins and outs of our data before we choose and select a variant caller. But in our case, we used the Freebase software for our activities.